And so next we have John. John, are you here? Ah, there he is. Great.
Good. All right, thanks, Jamie. Really glad to be here. This is my first time at this meeting, and I learned a lot. And I also have seen a lot of synergies and excited by a lot of what I've seen. So I'm going to talk about immune set point and immune health in my, and broadly speaking, my lab works on systems human immunology, where we measure individuals in populations and try to understand their responses to predict, and also then try to understand why. And then increasingly, we're interested in engineering, thinking about engineering cells. Yeah, you're welcome to take pictures of my talk. So one of the things I want to start with is variation, which I don't think I'm pitching to the choir here, that humans are variable. And you see that in immune response outcomes, like response to immunotherapy in cancer patients, and also response to vaccination, right? So you see that age is a big variable. But even if you look at younger individuals, in this case, like younger than 40, you can see a huge range of quantitative differences, hundreds of fold in terms of antibody response, for example, to vaccination. You also see that in the manifestation of autoimmunity. So in terms of the frequency and also the intensity or magnitude of disease flares for similar individuals. So for now, actually no general tools yet exist to answer questions like, did I respond to my last vaccine? Will I develop autoimmunity in the future? How will I respond in the future, right? So why do we have such variations in the response? So various factors like genetics and intrinsic factors like sex, and also depending on when we're looking at an individual, for example, during pregnancy, and also exposure and anything that we don't fully understand, we assign it to the microbiome. So together they kind of shape what we call a personal immune state. And this state can be quite vast in terms of configuration space, because for example, there are 10 to the 12 immune cells in the human body. And of course, the number of actual configurations and functional states can be quite relatively smaller, right? Because of constraints by evolution and so on. So now modern technologies, many of them have been mentioned here already, allow us to measure the immune system fairly comprehensively. So now routinely we can, from a blood draw or from a tissue biopsy, we can get 10 to the 5th to 10 to the 7 parameters. A lot of those data are coming from cells. So there's multimodal single cell measurements, and increasingly for tissues, we can get spatial information as well. We can take the cells out and stimulate them in vitro and look at responses. And we do transcriptomics, and you can also look at the DNA states through chromatin accessibility. And then there's also molecules. For example, you saw proteomics, and a lot of those are communicators among different immune cells, and some of them can be fairly long-lasting, and also metabolomics. So often the picture we see when we look at individuals in terms of measuring these immune state variables, we often see a picture that's more like the upper left, where you have reasonable temporal stability actually within a person. And in some cases, this can last over years even. Actually, you'll be surprising, a lot of these actually may not be impacted by age as much, some of them. So the key question is then, if you have these different states to begin with in these people, right? Does it mean something? And can we start to use that information to predict what's going to happen in the future in terms of health trajectories and also in response to various perturbations? So with that, so one of the probes that we and others use a lot is actually a vaccine. So a vaccine allows us to probe people and look at their very early and acute responses. So in a way, in an in vivo experiment, you can look at the very early, within hours to days, you can look at the so-called innate, but it's not just innate immune cells. You can see actually a very systemic response early on. And then you can also measure them out to months and then follow up to see, for example, actually whether a vaccine actually may have impacted certain cells for longer term. So, so more than a decade ago, we published a study. It was quite surprising at the time that even after accounting for age, sex, pre-existing exposure to influenza, this kind of baseline immune state, we call that the immune set point, actually can predict the response to the vaccine. And we later on take the same signatures that we derive from that initial study and develop surrogates, in this case, transcriptional surrogates, and look at multiple other cohorts, and even looking at other vaccines like yellow fever, which is a vaccine that's actually a live virus given to the individuals. And it's shown here that this kind of set point variable, in a way, they reflect the environment under which the responses are orchestrated, right? They're informative in telling us why do some people respond more than others. And later on, we conduct a single cell analysis using CySeq to measure both surface proteins, but also transcriptomics within these single cells. And that starts to give us specific ideas on what is encoding that signature. So in this case, it's actually a network of cells that are, you got these PDCs, they're expressing actually more type 1 interferons and more activated even in clinically healthy individuals. They're sort of locking these people into a higher, more poised state at the baseline. So once you know what you want to target, you can actually now compress that. So we were able to compress that state information across these 3 different cell populations into about 10 transcripts that you can measure in blood. And now you can look at larger populations and look at more people, for example. So now, what about beyond classic immunity? By that, I mean, when you think about the immune system, it's this surveillance system with cells that are going around our entire body. And often I tend to think of it as a homeostatic sort of maintenance machine, right? So it's detecting specific stressors and information in tissues and respond correspondingly. So you would think that a lot of the immune cells actually have information about the health of our tissues and in general about the whole system. So, and that's also why, of course, when you look at the immune system's involvement in various conditions, I'm biased, but I think you cannot find one single condition where you cannot find the involvement of the immune system, from neurodegeneration to pain and so on. So that begs the question, what exactly is a healthy immune system? What is a good immune system? What is immune health, right? So to look at that, one project that I want to highlight here that actually allow us to start to look at that, it's a project we call the Pan-Monogenic Project. So the idea for this project was that there's a lot of rare human conditions. They're typically mainly caused by a defect in a single gene, and they're like a mouse knockout sort of situation or knock-in. And so typically these are rare and they're studied in isolation. So about 10 years ago, we decided to talk to our colleagues to see if we can pull all of them together, but look at all of them at the same time. One thought it's actually using these natural perturbations to understand the underlying connectivity of the immune system better. The other one is to ask this question of, if you have all these very diverse perturbations to the immune system, and if you also have like clinically healthy matching individuals, can we actually study common deviations or what the immune system is sensing in terms of the problems caused by all of these conditions? So in a way, in addition to learning from the healthy people, we are learning from lots of negative examples, right, of what health should be. So with that, we measure quite a lot from the samples, and some of them longitudinally over multiple years. And what we found, one thing I want to highlight, is that despite the effect size of these mutations and the penetrance of the gene, a lot of the variation actually is still at the individual level. So going back to the picture I painted in the beginning, right? These individuals, you can actually find 2 individuals. They may have the same kind of mutation, but they may be as different. If you look broadly speaking in their immune system, they may be as different as individuals across multiple conditions. So the individual is the unit. So with that in mind, we kind of took 2 approaches. One, it's totally unsupervised machine learning. So we treated each individual as a vector of numbers, capturing all of those phenotypes we measure. And then we try to use unsupervised learning to come up with a latent space to quantify the relateness of these different folks based on their phenotypes. On the right, it's a supervised approach. We ask, can we actually come up and compute a probability of someone being healthy? If we got all these different types of sick patients, and then we got the matching healthy controls, right? So we can reduce the dimension down to 1 axis of a probability of someone being healthy. What was surprising is that if you take the first major axis in that latent dimension on the left, there are multiple ones. So if you take the first one, that explains the most variation. It corresponds really well to the probability of being healthy axis on the right? And then we actually did some perturbations experiments and asked, if you remove all the healthy subjects from the data set, so now there's no longer any clear contrast between patients and clinically healthy individuals, what we will get using the unsupervised approach. And the answer was that it was exactly the same thing. we came up with exactly the same first latent dimension on the left, as you can see. And on the right, each dot is an individual, by the way. On the right, even if you remove all the sick people now and only look at the clinically healthy people and apply the same unsupervised learning approach, it's noisier, but in a way, you got back the same axes, right? So there's the natural variation across all of these people. It's encoding in a way. We call them the immune health metric. It's the extent by which an individual is basically, it's immunologically healthy or not. So these are some of the parameters in the immune health metric. And I want to highlight NK cells as one of the ones that was really interesting. The count or the frequency of NK cells is a major component. The other one is CRP, it's in there, but if you regress out CRP and some of the inflammatory markers, the information doesn't seem to get lost. So a lot of what I'm going to show you next still applies. So one thing so this is actually how that metric, if you project them down on the X, that's the immune health metric. On the Y are the different patient conditions, populations. So HC is the healthy control. As you can see, the healthy control span a huge range, actually. Some of them are overlapping with the patients, right? So what does that mean? So we noticed in our own cohorts that that metric in the clinically healthy people, they correlate inversely with age, with chronological age, actually. So that was, it's not because of the structure of the data, like these matching healthy people are actually age match with all the disease controls. So, and I also show you that even if you remove all the patients, you all the healthy controls, you can still get this kind of metric. So, and one thing that we then did a meta-analysis of multiple vaccine studies involving older people, and we found that this metric, it's associated. The effect size is not that large, but it's associated significantly with the vaccine response in older people. That's one. The other one is we tested in Luigi's healthy aging cohort. And on the left, so we derive a proteomic surrogate. And so again, no additional training, just applying the same exact metric that we derived from our own cohort. And you can see the negative association being replicated. But you can see a huge, of course, residual, right? So the same 80-year-old, they have very different immune health in a way. And some of those can be attributed to BMI and so on, for example. And we also apply it to other data sets, for example, heart problems. And you can see that it tracks sort of the extent and the chronic nature of heart failure. And also it tracks responses to, in this case, RA patients, the responders, as you see that their immune health are rising significantly more than these non-responders. So again, I'm preaching to the choir here. I think you all sort of appreciate this concept where typically we look at folks who are clinically healthy and we look at pathology, but then there's a lot of hidden states in between. And I'm arguing that the immune system is a really good sensor for sensing a lot of these intermediate states, and especially diseases that take years to go from left to right, right? And I also don't want to oversimplify. I think it's high dimensional. Here we're just getting at the beginning of being able to project it down onto a one-dimensional metric. And then I wanted to also mention this effort that we have started. It's called the Zuckerberg Biohub New York. It's motivated by the concept I just mentioned, in that a lot of the immune cells are already sensing what's going on in our body in various organs, but we actually don't have a way of reading them out because we don't understand that code, basically, right? So imagine if you actually could bioengineer cells to do exactly what we want to read out. We actually send them to specific organs and be able to collect information in those organs as cells. And they can write information, for example, back to DNA, and through other means they can record. And we can now take that information to act on it. And initially, we probably need to act externally, but eventually we think that we can also program these cells as actuators, so they can become mini devices, right? So we have a fairly long runway with this. So the John Zuckerberg CSEI funded us for at least 10 years with the potential to be extended to 15 years. I'm mentioning this because I think this may overlap and synergize with a lot of what you are trying to do. And so if you have any ideas for collaboration, please reach out. And so the other one that's also highly synergistic with a lot of that's going on here, it's the Human Immunome Project. So my colleague Shai Shenoy and I are leading the scientific effort. And so our goal is to look at the world's immune system, and in sort of two phases. In phase one, We're in discussion to set up about 10 sites around the world. Each one, we're going to enroll about 1,000 to 1,500 people. And then we monitor them longitudinally. And in the interim, give them vaccines to probe them, just as I mentioned. And then with that kind of data, we think we can then scale it down to create a much cheaper way of measuring the immune system. So initially, we're going to measure everything as much as possible, but we're going to scale it down to create this immune monitoring kit. Based on data we have so far in the kind of studies I mentioned, we think that we can get it down to measuring several thousand things, a mix of proteins and transcripts and some cells. So that allows us to scale up to look at a lot more people by creating a lot more sites. So one of the first challenges is actually we want to create a site model to allow us to replicate these kinds of measurement and analytic approaches across the world to both build capacity, but also to do science together. So again, if this rings a bell, please contact me. So thanks a lot, and happy to take any questions.
We are tight on time, so one very short question, if there is one. Oh, there's our timer. If no short question, then we'll move on to our next. Thank you so much.